Hello everyone, welcome to Modern Biotech YouTube channel. And from today onwards, we are going to start one video series which is based on what is stem cells and what is the application of these stem cells. And also in this video series, we are going to cover what is stem cell therapy and what is the benefit of this stem cell therapy. So let's get started. So before starting this video, we need to understand what is a stem cell. So in simple definition, stem cells are the undifferentiated cells which has the ability to differentiate into other types of cells in the body. Okay, these cells are generally undifferentiated, present in undifferentiated form, and it can be differentiated into any type of body cells like nerve cells, muscle cells, by which if any damage is happening in our body, that can be taken up by these stem cells or that regeneration can be happened. Okay. And then researchers believe that stem cell based therapies they can be used for treat serious illness such as paralysis, Alzheimer's disease, Parkinson's disease, and moreover diseases like different diseases that we are going to discuss in the second part of this video series. And then after the understanding of the introduction about stem cells, we need to understand what is the type of stem cells. So stem cells are mainly divided into two types. First one is embryonic stem cells and adult stem cells. So first we will discuss about what is embryonic stem cell. Embryonic stem cells, as the name suggests, embryonic stem cells are those types of stem cells which is isolated or which is formed through embryo. So, in embryo, during the stage of blastula stage or blastocyst stage, the embryo containing a cellular mass that is undifferentiated in nature. What researcher will do, researcher will isolate that cellular mass and go on that cell culture based methods, and it can be differentiated that stem cell line can be established, or it can be used after the establishment of stem cell line that can be used for multiple purposes like generation of some organs or some special type of cells in a for the human body. Okay. And ESC is a very valuable tools for the understanding the complex mechanisms involved in the development of specialized cells, and also the establishment of organ structures can be overcome by this using ESCs. And also ESC are the pluripotent cells which can give rise to different types of or any type of somatic cell types. Okay, and these are naturally present in the embryo. That what we are doing, just we are taking that cells we are isolating that cells from the embryo, and we are culturing that cells in a cell culture based medium, by which the stem cells can be generated. This is a pictorial representation which is showing that how the stem cell can be generated using embryo. Okay. As I told you earlier, this is the embryo at the blastula stage. These are the cells which is containing that undifferentiated cells. What we will do, we will remove this layer and we will take this cells and grow on the cellular medium. Then you will, once stem cells are developed, then using the different differentiation factors, these stem cells can be differentiated into different types like ectoderm, mesoderm, and endoderm, and after that it can be converted into skin cells, nerve cells, and many more. This is the mechanism how that embryonic stem cells can be generated. So after the understanding of embryonic stem cells, we will move to the second type of stem cell, that is adult stem cell. Adult stem cells are mainly two types. First one is the stem cell which is present in fully developed tissues like brain, skin, bone marrow. So, but in these types of stem cells, one drawback is there. What drawbacks? Because these are isolating from a certain type of tissues, so it can only generate a certain type of cells only. For example, if stem cells are isolating from liver, it will generate a special type of liver cell only. That is the drawback of this type of adult stem cells. And the second type which we are going to discuss is a very important type, and it is very important also, like iPSC. What is iPSC? Now, in iPSC means I stand for induced pluripotent stem cells. That means these are the normal cells that are converted into pluripotent stem cells. So, one thing will be coming in your mind, how we can convert a normal differentiated cell into the pluripotent stem cell. So, firstly, these type of experiments done in 2006, and the mechanism which they are this researchers are using to converting the normal somatic cell to the stem cells are overexpression of transcription factors like OCT4, SOX2, TLF4, and CA. These are also called Yamanaka factors, and these are the factors which are expressed for maintenance of the stem cell nature of any stem cells. So, or in simple word, we can say that these are the four transcription factors which is responsible for the maintenance of undifferentiated nature of a stem cell. So, if the researchers thinks that if we artificially express these factors into normal cells, it can convert the normal somatic cell also into the stem cells. So they got a positive result also in this research, and uh, in human cells, if in human normal cell, if we express these four Yamanaka factors in that somatic cells, it can be converted into the it can be converted into the stem cell that will come as iPSC. And the one more benefit of this iPSC is when we are using ESC means embryonic stem cell, it has one drawback. The drawback is because we are using the embryo, so these are the unethical for the research. That's why you production of iPSC, because these are generated from gametic cells. So no embryos can be used. So therefore no life can be harmed. That's why it's comes under ethical issues. So it's better or like it's more better or more useful than ESC. And also one more benefit of these cyber cells, like if we are having any somatic cells, like if we are having any cell lines of any particular organ, we can express these four Yamanaka factors in this cell line, and if it after this expression of these four factors, this cell line can be converted into stem cells. So with the help of this technique, with the help of introduction of these four Yamanaka factors, any somatic cell lines can be converted into stem cells. This is the easiest way of conversion of these somatic cell lines into stem cells, and also uh, more stem cells can be generated. The benefit of this iPSC is it can be used for the generation of different kinds of cells and tissues. These are more benefits. And now also after discussion of these types of cells and what is stem cells, the one thing will be coming in your mind: what is special in that stem cells which make it more interesting? So in this things, mainly three important things are there. That is, first is it increases the understanding of how disease occurs, because we are using the cells which is generated from human only, the stem cells. With the help of that, we can see how the disease are working in the body, or how the disease are affecting our cells, by which we can easily understand the mechanism of the disease, and drug can be generated for that mechanism, that the disease. And in the second way, it can be used to generate healthy cells that can replace the affected cells or affected cells or damaged tissues. Okay, this can be called as regenerative medicines, and it can also be used to test the new drugs, and to to see the toxicity of the drugs and see the safety and effectiveness of that particular drugs. Okay, these are the three important things why the stem cells are important, and after these things, we need to understand what will happen to the stem cells. The stem cells will go, after these all things, the stem cell can undergo in four ways. First, four stages, like first it can maintain in quiescent state, quiescence, and second is symmetrical cell removal and asymmetrical cell removal, and symmetrical division without cell removal. This, these all states we will understand using the diagrammatical representation. In this one, we can easily say we can easily understand that quiescence means that stem cells which we are having, it will remain in that form only. and it will present in bone marrow, and it will remain in that undifferentiated stage, undifferentiated stage until or unless when differentiation factor will come and it will convert to other different type of cell. In second type of phase, symmetrical cell removal, what will happen? The stem cell will convert it into stem cells. It divide into stem cells and it generate two stem cells, by which the number of stem cells can be generated in a particular type of tissues. This is important to generate a number of stem cells in any tissues. Okay, and then in the third type, asymmetrical cell removal, in which the stem cell will divide into one stem cell and one differentiated cell. This type of phase is needed for the type of tissues where any damage tissues need to replace using that. So the stem cells can be differentiated into the particular type of cells and it can be replaced. Other regeneration can be happen. And also because it's generating a stem cells also simultaneously, so the stem cells number can also be maintained. In the fourth type of fate, a symmetrical division without cell removal. In this type of fate, a stem cell will generate two type of differentiated cells that can be used for generation of or that can be used for multiple uses in a body. So, whenever the stem cells are present in our body, it will go into these four type of fates. So, after reading of these all things, the basic of stem cells, one thing will be coming in mind. What are the challenges of in the stem cell research that researcher can face? So first of all, the first challenge which researcher faces during the embryonic stem cell or any stem cell research is ethical issues, because embryonic stem cells are unethical because we are using an embryo that is equal to the life or young one's life. So because we are using embryo, it can it can cause the death of that embryo. That's why it will become unethical for the use of any purposes. But because it has more benefit compared to the ethical things, that's why sometime when ESC when stem cells are needed, we can use ESC but not too much. That's why here the scientists will overcome this drawback by using the IPSC that we discussed earlier on. Then after the development of stem cells, we need to differentiate that cell. So for the differentiation, we need differentiation factors. So we need to understand the different differentiation factors that can be used to differentiate a particular stem cell into the particular type of cells. Okay, and the another challenge is that embryonic stem cells can be rejected by the body of a human. So we need to overcome this challenge because we can develop a stem cells, but if that our body is not accepting that stem cells, so no use. That is a problem. And also it can generate an immune response also, immune tolerance also. Okay, which that can be harmful for the human being or that patient. And then the fourth challenges that any researcher can face is adult and pluripotent stem cells are very hard to grow. So researcher need to look the different ways by which we can easily grow these cells. Okay. So for this video, uh, we will come in this video we covered all the basic of the embryonic stem cell. If you are having any doubt regarding these stem cells and basics, you can ask me in the comment section. So we will meet in next video in which we will talk about what is stem cell therapy and what is the different application of stem cells. So thank you for this video. Thank you for your attention. So we will meet in next video.